So good afternoon. It is noon time here on the East Coast. My name is Kate Fitzgerald. I'm at UMass Chan Medical School, and I'm delighted to be one of the hosts of the Global IMMUNOTALK. So I just want to remind everybody that next week we have the last speaker of 2023. That will be Tim Hand, who's going to join us December 13th. But for today's talk, we have Professor Xiaoyu Hu, who's joining us from Beijing in China. Xiaoyu, is professor at the Institute for Immunology in Tsinghua University in Beijing. And I'm just going to start with a brief introduction to her previous record. So Xiaoyu received her Bachelor of Medicine degree from Peking University and then her PhD from Cornell University, working with Lionel Ivashkov. And she's then gone back to China. And as I mentioned, she's currently professor and director of the Institute for Immunology in Tsinghua University. So her research really aims to better understand transcriptional, translational, and metabolic mechanisms that regulate macrophage and other myeloid cell responses to microbial and inflammatory cues. She is the recipient of multiple awards, including the Christina Fleischmann Award to Young Women Investigators from the International Cytokine and Interferon Society. She's also the recipient of a Distinguished Young Investigator Award from the National Science Foundation of China, and the Newton Advanced Fellowship from the UK Royal Society. She serves on numerous editorial boards, including Trends in Immunology, Science Advances, eLife, and Mucosal Immunology, and she's co-organized a number of important meetings in the field, including Cell Symposia, the International Congress of Immunology, and the Society for Leukocyte Biology annual meeting, as well as a Cell Research Symposium. So she's a strong advocate for women in science, and has really sort of had a really impressive trajectory in her career so far. And I will point out, it is 1 a.m. in Beijing, so I'm really impressed, Xiaoyu, that you're still awake and ready to give a talk. So before we get started, I wanted to. We usually, because we have such a broad audience from around the world, and particularly a lot of early career scientists, students, and trainees, I think people are always really interested to hear a little bit about the famous scientists who speak in this global series. So I was wondering if you could tell us a little bit sort of about your own kind of history, and in particular, I think as an immigrant yourself, what kind of advice would you give to a young scientist who's training abroad? You can draw on your own experience from your time in New York.
Yes. Oh, thank you, Kate, for that very flattering introduction. And by no means I'm famous, and just a struggling immunologist trying to understand how myeloid cells work. And but I think the case question regarding how immigrant scientists feel and function in a foreign environment, I think is really pertinent to my own experiences. I actually moved to New York City right after I obtained my bachelor's degree, so I was pretty young, and so of course it's a very dazzling world, but I think if I draw from my personal experiences, and I think the only thing I will say is that don't be intimidated by anything you see or anyone that give you a hard time. And there are certain people are privileged to be born in certain places, but that doesn't mean they are smarter than you or they have a more fulfilled life than you. So just be very certain to pursue your goals, and I'm sure things will come along. So I think my only thing is don't be intimidated, and just believe in your capacity and your goal, and you'll get there. And all this things that seem pretty intimidating or dazzling at the beginning. And you will find out they are just what they are.
It's so true. Yeah.
Yeah, so I think that's not sure how helpful that is, but kind of that's my 2 cents on
Yeah, no, I think that's very
immigrating to a
nice.
very different place. And I think one thing I really like to see is, if somebody, has the courage of immigrating to a completely different country and starting a completely new career, and you already like beat 99% of the population, right? You're great. And
Yeah.
you
Yeah,
can.
I agree. I'm always impressed with people who are willing, and I like you, I did the same thing. I did all my training in Ireland, but to sort of take that step to move to another part of the world, leaving your family and friends behind, it's very brave. I think it's a good sign of resilience, which you need a lot of in science, resilience and kind of perseverance as you progress through your career. Yeah. So good. That is great advice for our trainees. Okay, let's get started. So if you can share your screen, and then you put that into presenter mode, and then I'm gonna turn off my camera just so we can preserve some of the
Yeah, can you see it well?
signal. Yeah, that looks great. Wonderful.
Okay, great.
Okay,
So I will
wonderful.
start. Thank you
Yeah,
again. Yeah.
thank you so much.
Yeah, sure. And okay, I will get started. So it's really my pleasure and to be on this Global IMMUNOTALK. And I have been following the Global IMMUNOTALK Twitter account for a while. So finally I'm here. And so I am on Twitter. So also, feel free to ask me questions, and even during the talk. And so I can respond to all the questions afterwards. And yeah, so as Kate introduced, and I'm from Institute for Immunology at Tsinghua University, and I received my Bachelor of Medicine degree from Peking University, and then I moved to New York City to pursue my PhD. And I think my lab, for the past 13 years, my lab has been working on how myeloid cells actually respond to various cues, and the current research directions mainly focus on macrophages, and we're trying to understand the molecular mechanisms governing macrophage activation, the biology of tissue-resident macrophages, and more importantly, how monocyte macrophages contribute to human autoimmune and inflammatory diseases. And yeah, basically I think this the starting slide sounds really simple, and I just want to make an also equally simple point that the production of the immune effector molecules do follow central dogma, and sometimes many people forget, like immune cells are also cells, and they follow central dogma strictly. And for example, I draw a macrophage here, and macrophages receive signal inputs and from various sources, and they really respond to such signal inputs by producing immune effector molecules such as chemokines, cytokines, and various products. And the production process, right, actually exactly follows central dogma. We do need transcription to get mRNA, and again, we need a translation to get protein. And what I'm trying to stress today is that oftentimes we focus so much of our attention on this step, transcription, right? So we know a lot about how macrophages, all other immune cells, respond to environmental cues through modulating their transcriptional processes. But in contrast, we know so little about how they respond, right? They modulate their translational processes. So yeah, very recently, my PhD student Xinxian Liu and I wrote a invited review in Advances in Immunology book. And so basically in this review, I think if you are interested, feel free to read it. I think it's pretty interesting, and we're trying to outline the post-transcriptional steps and in myeloid responses to inflammatory cues. And actually, if you think about it, and the transcription process is extensively studied, right? We know so much about it, and what are the cis elements, what are the trans activating factors, and what even the epigenetic factors, the chromosome looping, and all kinds of regulation at all kinds of different levels. But okay, in contrast, there must be some level of regulation at the translational level, right? If we just draw these two processes in parallel, but okay, but if you think about it, do we really know what are the cis-like elements? Do we really know what are the trans-like factors? We actually don't. And also transcription, we know a whole lot about how specificity is achieved. Of course, like we will think about transcription, we are not going to transcribe all the genes in macrophages all at once, right? There is very strict specificity. But in translation, we know very little about how specificity is achieved. I think similarly, we're not going to translate all macrophage mRNA into protein all at once. In macrophages, there are about probably 10,000, approximately 10,000 genes expressed in resting macrophages. Certainly, we're not going to translate all mRNA, and there must be specificity of translation, but we know, again, we know very little about it. So basically, I think this is really the process, and I'm going to talk to you today, and I will give you a few examples and how translation process can be regulated and to actually critically influence the outcome of immune and inflammatory responses. I'm going to give like 3 little examples, and I'm going through the first 2 examples very quickly, and I will get to the last example. I think probably the most important example that we can actually harness the translational regulation to for the benefit of therapeutically targeting human inflammatory diseases. And yeah, this example actually came rather long time ago, and at the time, and my lab was studying how Notch signaling regulates macrophage inflammatory responses. So basically, we observed that the Notch signaling positively regulates the macrophage inflammatory polarization and effector function. So in the absence of Notch signaling, and macrophages make less amount of cytokines such as interleukin 12 and less really key enzymes in defense, that is NOS2. So, but how does this work, right? And yeah, this is a question mark. Basically, how Notch signaling controls the production of these inflammatory mediators. And at that time, we observed that Notch signaling actually controls the abundance of a critical transcription factor called RF8. So in the absence of Notch signaling, there's reduced amount of RF8, and RF8 is one key transcription factor driving the production of the factors I just mentioned, such as IL-12. And however, during the process, we found that RF8 actually RNA level is not really altered by Notch signaling. So then it's really mysterious. Okay, then how RF8 is regulated by Notch signaling, and this is the experiment actually I did and as a bench and with my postdoc, and we really labeled newly synthesized L8 protein with S35, and you can see upon LPS stimulation, and actually the synthesis of L8 protein increased quite a bit. And however, in the absence of Notch signaling, such induction of L8 protein synthesis is completely abolished. So basically, this very simple experiment tells us the Notch signaling controls L8 protein synthesis, right? That is very likely at the translational level. Yeah, so without going into the details, and basically this data give us hints that, L8 is regulated at the translational level by Notch signaling. And this is like one example, like a long time ago. At that time, it made me very puzzled. Okay, how does this work, right? To be honest, we still don't have clear-cut answer today. But this is clearly biologically very important, and this really get us intrigued to study how translation contribute to inflammatory responses. I think the second example actually comes from a very interesting crosstalk between our nutrient sensing system and our host defense system. And because nutrient sensing, the front line of nutrient sensing really occurs in the intestinal epithelial cells, so we want to study this crosstalk in intestinal epithelial cells. And we starved the mice for for various time points and did a very simple experiment and to test the expression of intestinal epithelial cells expressed genes or proteins. So of course we did a lot of profiling and blah blah blah, and basically we found that there's a protein called HAS1 and abbreviated for hairy and enhancer of split 1. So HAS1 is a transcription repressor, and this is basically the intestinal tissue we took from the fed mice and from the mice that had been fasted for various time points. And I think you can see like a very strikingly after fasting, the HAS1 protein level went down in intestinal tissues. However, the HAS1 RNA levels did not really change, right? It's remained constant throughout the fasting process. Again, this prompts us to think about, okay, this regulation actually must happen at the post-transcriptional level, and could it be protein synthesis yet again, right, for controlling HAS1 protein expression? And so to really test this hypothesis, we perform the ribosome profiling of intestinal epithelial cells, and those are the primary intestinal epithelial tissues, and fed or fasted. And this is from the ribosome profiling track of the HAS1, right? We can clearly see that the translation efficiency, short for TE, and it's clearly decreased in the fasted mice compared to the fed mice. And this tells us that the HAS1 translation actually is controlled by nutrient availability. And this is like actually the primary tissue taken from in vivo. I think this data is pretty convincing, telling us, in vivo it's really happening that the HAS1 protein is actually controlled at the translational level. And okay, what's the mechanism for such translational regulation? And of course, like probably the first pathway that came to somebody's mind is mTOR. And here we look at the mTOR pathway activation, and this is basically the HAS1 staining. The brownish color actually indicates the HAS1 positivity, and in fed mice there's lots of HAS1 in the intestinal tissue, and when we fasted the mice, of course, this HAS1 protein level decreased. But at the same time, the phospho-4EBP1 actually also decreased, actually probably with very similar time course as HAS1. So this makes us wonder, okay, maybe the mTOR and the 4EBP signaling is controlling HAS1 protein translation, and thus the abundance of HAS1 protein. And therefore, in vivo, we treat mice with rapamycin, and rapamycin actually indeed blocked, of course, phospho-4EBP, but also it decreased the HAS1 expression. And however, the rapamycin did not decrease the HAS1 RNA, right? I think this actually supports our hypothesis that the HAS1 protein is translationally controlled, and probably by mTOR signaling. And so what kind of signals, nutrient signals, the mTOR signaling is sensing to control HAS1 expression? And here is basically we perform the in vitro experiment to figure out exactly what kind of nutrients are required. Basically, we add all different kind of nutrients and to the starved cells, and we can see that the amino acid, right, the mixture of 20 amino acid can fully restore HAS1 expression. And also we want to see, okay, do we need all 20 amino acids to restore HAS1 expression, or certain amino acids actually are sufficient? So my student Shaonan Liang actually took really a rather comprehensive approach, tried 20 amino acids one by one and all kinds of combinations. What she found is that arginine together with leucine can largely restore HAS1 protein expression as well as 4-EBP phosphorylation. So therefore, the arginine and leucine are likely sensed by the mTOR signaling to regulate HAS1 protein expression. So yeah, so without time to going through all the data, so basically our model is that in the nutrient deprived condition, so basically when the mice are fasted, and so insufficient nutrients actually sensed by the mTOR signaling, and therefore the mTOR signaling activity actually through a translational mechanism results in decreased expression of HAS1 in intestinal epithelial cells, in particular in Peynian cells. And remember, HAS1 is a transcription repressor, so therefore decreased HAS1 expression actually boosts expression of the HAS1 target gene, and one of such target genes is ATOH1. So ATOH1 itself is a transcription activator. So ATOH1 actually drives expression of certain genes that are required for maintenance of the antimicrobial defense during the nutrient deprived conditions. So this is a feedback loop, and to maintain sufficient host defense under nutrient deprived condition. And if this loop is disrupted, and the mice are actually highly susceptible to a bacterial infection and under nutrient deprived conditions. So yeah, so basically I think Shaonan also draw a picture, and we have a cartoon basically saying this is a robot and that doesn't need to eat, right? So this is basically the mechanism of antimicrobial defense, right? Even without food. So this robot can still fight bacteria, fight bugs, and even when the food is scarce. Yeah, so okay, so those are two examples of we did most of the studies in mice, and it's lots of mouse genetics and knock-in, as usual. But what really matters is in humans. So basically, the next I think majority of the rest of the talk, and I'm not going to deal with mice a lot, and we're going to talk about data from patients. And so this is a collaboration, very close collaboration done with Professor Wei Donghan from the Department of Biotherapeutics and from the Chinese PLA General Hospital, because the translational studies cannot be possible without collaboration with clinicians. And again, I would like to really highlight two contributors from my lab, graduate student who just graduated, and Yu Zhuo Yang, and another MD student, Xiaoyan Xing, who is doing her clinical training now. Yeah, so in human, we try to understand a very serious inflammatory condition called CRS, cytokine release syndrome. So CRS is a major adverse event associated with CAR T therapy, and this review basically summarized the incidences of CRS from various clinical trials. So we can see the number here is on average, I think 70 to 80% of patients receiving CAR T therapy would develop CRS, and the CRS actually can be life-threatening, right? It's can really cause patient death. So it's a very serious side effect that has hampered the wild application of CAR T. So, however, the mechanisms of CRS is now well understood. So recently, we also wrote a review and actually an opinion article with Xiaoyan Xing in transient immunology. We're trying to really synthesize our knowledge of what are the risk factors for CRS development. Basically, there are three questions, right? So who would develop CRS? Not everyone would develop CRS, and when CRS would be developed and how CRS is developed. And basically, after reviewing literature, and we still don't know much about the mechanisms of CRS occurrences and how we can prevent CRS. And again, if you're interested, you're more than welcome to reading this article. And so how do we manage CRS clinically? And currently, I think there are really two kinds of therapies, and one is corticosteroids, and typically dexamethasone will be applied, and another is L6 receptor blockade, because L6 is a major driver of CRS in human conditions. And for these two therapies, and as we can see, those are well-established anti-inflammatory agents used for other disease indications. They are approved for CRS, really based on empirical clinical data, and not really from the rationally designed clinical trials, or really a CRS specific therapy. So those are really for general inflammatory conditions. And so basically our study with CRS really began with an accidental clinical observation. So basically one day there's this patient and receiving CAR-T therapy. This CAR-T is a CD19, CD20 so-called tandem CAR-T, and that targeting B cells. And the patient received the CAR-T, and at day 6, this particular patient developed certain cardiovascular symptoms, and including ventricular contraction and other symptoms that needs medical attention. And the clinician that took care of this patient, right, she gave the patient actually oral one dose of oral metoprolol. So metoprolol is a beta-1 blocker that used to treat cardiovascular conditions. And I think this clinician basically observed actually a rather bizarre phenomenon, that is, after this one dose of metoprolol, the patient temperature, right, because this patient have undergone CRS, so has very high body temperature, the body temperature actually dropped immediately, and also the serum L6 level actually dropped. And so this really prompt us to consider, okay, could metoprolol be used to treat CRS? So what is metoprolol? Metoprolol, as I said, is a selective beta-1 blocker, and it's being used, very widely used to treat conditions like high blood pressure, chest pain, and it's one of the top 20 genetic drugs worldwide, and it's also on the WHO's list of essential medicines. And in US alone, the annual prescription of metoprolol exceeds 70 million, right? So basically, it's a very widely used medication. It's very safe, and it has been prescribed a lot. So we want to see, okay, can we repurpose metoprolol for treatment of human CRS? And I think there are two critical questions that need to be answered. I think first one is the mechanism of action. The second one, of course, is the clinical efficacy. So for the mechanism of action, because we are dealing with human patients, and so we need to develop an in vitro system to study CRS. So here we devise a system of taking human CAR-T and killing human B cells, and then taking so-called killing supernatant and incubate human primary CD14 positive human monocytes isolated from peripheral blood, and with the killing supernatants. And we want to see, okay, can this process actually mimic CRS? And I think it can, and this is basically the serum L6 levels, right? In vivo in CAR-T in patients receiving CAR-T. And this is basically the IL-6 protein levels in our KS-induced CD14 human monocytes. And we can see the kinetics of in vivo and in vitro IL-6 production actually aligned very well. So we think we do have an in vitro system with primary human monocytes to mimic CRS. And we were also very happy to see that in this particular system, metaprono actually consistently and robustly blocked IL-6 production. I think one line represents one blood donor, so actually it's very consistent among different blood donors. However, and to our surprise, metaprono did not block IL-6 mRNA, right, in all these donors, and clearly metaprono did not achieve a consistent trend of blocking mRNA. So this leaves us with a possibility that metaprono blocked IL-6 production at the post-transcriptional level. So we rule out lots of possibilities, like metaprono did not really regulate IL-6 protein stability. So we were left with the possibility that metaprono might regulate IL-6 protein translation. So to test this possibility, we performed ribosome profiling of human primary monocytes. So yeah, basically we took human primary monocytes cultured with KS killing supernatants and in the presence or absence of metaprono. And I think the first thing is we saw that the metaprono did not really alter the global translatome. So the global translatome is pretty normal. However, upon closer examination, we did see that metaprono downregulated translation efficiency of 125 genes, and those 125 genes actually clustered to the inflammatory gene category. And we were happy to see that IL-6, right, is among the 125 genes whose translation efficiency is downregulated by metaprono. And we can take a closer look at the track of the gene track of IL-6. And this is mRNA expression, and the metaprono clearly did not inhibit IL-6 mRNA expression consistent, right, with our previous data. And however, it clearly blocked translation efficiency of IL-6. And again, this actually supports the notion that metaprono indeed blocked translation, right, but not transcription of IL-6. And translation is a highly regulated process. And I know there are so many reviews about transcription, but translation is equally fascinating. And translation has multiple stages as well. And similar to translation, it has an initiation stage, elongation stage, termination stage, and then ribosomes recycle. So we want to further investigate what are the exact mechanisms by which metaprono blocked IL-6 translation. And we saw that actually metaprono preferentially blocked the ribosome profiling signals at the IL-6 gene body region. So we devised index called elongation efficiency index, and to really indicate the elongation process versus the initiation process. And we can see that the prophylactic treatment as well as the conditional treatment of metaprono can block elongation efficiency, but did not really impact the initiation process of IL-6 translation. So this tells us, okay, elongation is very likely to be targeted by metaprono, but exactly how? And elongation actually is very complex, as I recently learned, and it's controlled by really multiple factors, and I think among which there is a factor called EEF2. Okay, EEF2, the active EEF2 is actually unphosphorylated, and it can promote elongation very well. However, the phosphorylated form of EEF2 is relatively inactive, and it's not very good at promoting translation elongation. So basically here we see that metaprono can boost the phosphorylation of EEF2, which means actually it hampers translation elongation, right? And there's this enzyme upstream that can phosphorylate EEF2 to the phosphorylated form of EEF2. It's called EEF2K. So metoprolol actually can reduce the phosphorylation of EF2K, and which we believe, without getting into the details, and we think metoprolol controls the activity of EF2K and the EF2 axis to regulate IL-6 translation. So then what's upstream of EF2K? And again, we look at the RNA profiling data, and it's very, I think, obvious that there are two enriched pathways that are downregulated by metoprolol, the RAB1 signaling and cyclic AMP signaling. And there's this gene called RABJEF3 encoding a protein named EPAC1. So RABJEF3 is highly consistently downregulated by metoprolol. And if we knock down RABJEF3 in human primary monocytes with RNA interference, and we can reduce IL-6 expression. And furthermore, in the RABJEF3 knockdown cells, the metoprolol no longer inhibits IL-6 expression. So therefore, somehow within metoprolol downregulate the expression of EPAC1, and the EPAC1 sits upstream of the EF2K, EF2 axis to control IL-6 translation. And that's about the mechanism. Of course, it's not complete, but somehow we so provide evidence that it's a very likely target the IL-6 translation process. Okay, but the $1 million question is, okay, is metoprolol really effective at alleviating CRS, right? Because we only had this one patient. So basically, together with Professor Han, we launched an investigator-initiated phase 1/2 clinical trial and to evaluate the safety and efficacy of metoprolol on CRS. So initially, we assessed 26 patients, and finally 15 patients enrolled in this study, and plus the original, the patient number 0. So in total, we have 16 patients. Yeah, I think the first goal of the trial is to evaluate the efficacy, the safety actually, of the metoprolol on CAR-T therapy. And this drug, as I said, is very widely used, and it has like millions and millions of prescriptions annually. So it's ought to be safe, and this is indeed what we see. Actually, this drug did not really alter all the vital signs of the patients receiving CAR-T therapy. And I think another concern is that, okay, this drug may be good at dampening inflammation, but would it also actually reduce like CAR-T proliferation, right? So if this is like a general immune suppressing drug, like dexamethasone, you would really hamper CAR-T proliferation, then it will be not that useful for controlling CAR-T-induced inflammation, because it will kill CAR-T. And so to really test this possibility, we took the bone marrow aspiration, I think, from the patients before and after metoprolol treatment, and performed single-cell sequencing analysis of the bone marrow of the same patients, so we can actually see what's the effect of metoprolol, right? And basically, we can see it did not really affect CAR-T expansion. So CAR-T actually cells expanded really well in this particular patient, and it's really the dominant, I think, population of the bone marrow at some point, and also it does not affect the killing of B cells. Basically, the B cells are very effectively eliminated from bone marrow, and also it doesn't affect the population of monocyte macrophages. So basically, from all the patients, and we measure the peak CAR-T numbers in their blood, peripheral blood, and metoprolol did not affect CAR-T expansion. And Also, at the same time, the metoprolol did not affect the CAR-T response rate and also the progression-free survival rate. I think up to this point, and I think some patients have been alive for like more than 2 years after a CAR-T therapy, and I think it clearly did not have a negative impact on patient survival. And okay, is this really effective, right, at alleviating CRS? And those are patients, they have great degree of variability, and but so these are the individual patient data, and but you will pull them together, and in the 8 patients that are receiving metoprolol conditional monotherapy, which means the patient will be given metoprolol, I think, and the metoprolol only, I think, once they develop CRS, and this is actually very effective at reducing the serum IL-6 level and also reducing CRP level, as well as reducing body temperature. So I think from this data, I think we are pretty confident that metoprolol can relieve CRS. And I think one piece of data I showed you is that in vitro in our killing supernatant culture system, metoprolol actually did not affect IL-6 mRNA expression. Instead, it dampened IL-6 protein production. But is this the same mechanism in vivo? Actually, it's a very hard question to be answered, because we cannot really do so much mechanistic study in vivo, but we can get probably supportive evidence. In this particular case, we obtained the serum protein IL-6 level as well as the PBMC RNA IL-6 level from the same patients at exactly the same time point. For example, for this patient number 15, and the metoprolol is very effective at reducing serum IL-6 protein level, and as I showed before. And however, at exactly the same time point, metoprolol did not really alter IL-6 RNA level in PBMC. And so therefore we believe, although this is not definitive causal evidence, but this suggests that in vivo metoprolol might also regulate IL-6 expression at the post-transcriptional level, very likely to be at the translational. So from this all-human study, and basically we started with accidental clinical observation with metoprolol and relieving CRS, and we delved into the mechanisms, and we actually found out that metoprolol actually, with the data I didn't have time to show today, really acting at the beta-1 receptor and dampen the translation elongation of IL-6 through targeting the EF2K, EF2 axis. So the outcome of inflammation basically is that with the same level of IL-6 mRNA template, the metoprolol treated human monocytes actually produce less amounts of IL-6, right? So therefore, in the patients, it's manifested as relieved inflammation and the CRS. So so basically the take-home messages of this study is that it's all human-derived materials, and not a single mouse is killed in this study. And from all human study, and also from the clinical trial, and we know that the oral metoprolol is a very cheap and very safe therapy, can actually significantly alleviate CRS without compromising CAR-T efficacy. And I think most importantly, I think this study provides a proof of concept case for targeting translation of key inflammatory mediators as a safe and effective approach for controlling severe inflammation. And this is actually very different, I think, from all the mouse genetic studies I usually do in my lab. This is really a bedside to bench study, right? We have a bedside observation, and we took it to the bench, and we tried to dissect out the mechanisms of action of metaprono and to inhibit IL-6 translation. And but as a next step, right, we would like to take the bench to bedside again to complete the circle, and we would really like to understand the underlying mechanisms of CRS and therefore to rationally design CRS therapies. And yeah, next we are going to share with you some published data. I think really exciting ongoing research in my lab. So we're trying to understand actually what drives translation in CRS. And this is again the human system and comparing to the resting human monocytes. And actually we can see a global translational boost in the CRS-treated human monocytes, right? It's very striking, actually. This is the ribosome profiling data, and it's not subtle. It's something like when you treat human monocytes with clean supernatant, it's like this massive translational boost. And to really study this process, I think in human cells we are again certainly limited by the tools that are available, and we want to see, okay, can we replicate this in mouse? Actually, we can. We took murine primary bone marrow derived macrophages. We also treat those macrophages with killing supernatants. Of course, a little bit twisted. Those won't be human cells, right? So we saw a similar translational boost with a polysome profiling approach. And so I think this is all really good, and this will allow us to do something that we have been always want to do, that is the CRISPR screening I will talk about. And so this, okay, I think first of all, I think we really want to see in this particular system, okay, what are the relative contributions of translation versus transcription to the development of CRS, right? So in this case, my student Xin Xianliu took the ribotag mice and stimulated the ribotag mice, the bone marrow derived macrophages and with killing supernatant. And the y-axis basically designates the contribution of translation, and the x-axis designates the contribution of transcription. So it's a rather like complicated graph, but I will just draw your attention to the class 1 genes. Okay, class 1 genes means those genes, the translation is being upregulated by the killing supernatant, but the transcription is either not changed or downregulated, which means the translation is really the driving force, right, of enhanced expression of the class 1 genes. And if we look at the number of the class 1 genes, it's actually a lot, right? It's more than 2,000. It's a majority, basically, of the genes that are upregulated. It's actually driven by translation rather than transcription. And to better look at this, actually we can really define this is basically all the genes. If we think 100% is all the genes that are expressed in human macrophages, probably around 10,000 genes. And in a CRS response, about a quarter of them actually are upregulated in terms of translation efficiency. In contrast, in the LPS response, actually only about like 3.8% of the genes are upregulated in terms of translation efficiency, right? So immediately from this contrast, we see actually a very distinct program, translational program, that is specifically driven by CRS, but not by LPS, right? So here, transcription is actually not the telling factor between CRS response and the LPS response, and it's a translation, right? So yeah, so basically, of course, the next step is we're trying to understand what drives translation and hyperactivation in CRS. And I think here, Xinxian took the Cas9 transgenic mice and performed the CRISPR screening. And I think this is still work in progress. So basically, we identified, I think, quite a number of drivers, I think, in this case. And we use IL-6 as readout. And we can really, I think now focus our attention on some of the drivers of the translation process, and we really hope to find the major players in this process that is really minimum knowledge is known about. And again, we back to this cartoon in our review that the contrast between translation and the transcription. And I think one notion is that targeting translation give us a window of opportunity for damping bad inflammation while sparing good inflammation, right? That is the inflammation that's probably induced by LPS that's involved in host defense that we do not want to totally dampen. And I think, to just sum up, and I think I give you 3 examples and very brief examples of how translation step can be regulated in various cell types, and 2 of them are monocyte macrophages, and this particular example is in intestinal epithelial cells, right? So this steps can be critically regulated to influence the outcomes of immune and inflammatory responses. And I think the last example, particularly in human, right, we show that it can even be therapeutically targeted for treatment of human inflammatory disorders. And of course, I think there will be a lot more examples to come. And, I really hope, I think everyone considers, translation as a possibility. And when you come to study your favorite gene or pathway, right, and not just transcription. And yeah, here is my lab, and I think I already mentioned some contributors. And here is a student who performed the translational boost study, Xingxian Liu, and here is a student who graduated, Shaonan Liang, who performed the translational study in intestinal epithelial cells and of course the funding source. And last but not least, I would really like to do a little advertisement, and for the Cell Symposium in Mucosal Immunology that will take place in Beijing, and in May 15 to 17 next year. So I think it's a great symposium that has great scientific program, and also it's a wonderful season to visit Beijing. And I think the abstract deadline is January 5th, so it's coming. And I really would like to encourage everyone to take a look at the website and to hopefully submit an abstract and to come to meet us in Beijing. Okay, thank you.
That was wonderful, Xiaoyu. That was really a tour de force. Yeah, beautiful kind of detailed mechanistic work, but then I love translating that into human disease too. Excellent.
Yeah, thank you.
Very nice. Okay, I just want to, so firstly, thank you for joining us from Beijing. So late in the evening or early in the morning, which, it's almost time, it's definitely time for your bed. But I want to just remind people how to ask questions for Xiaoyu. This will be through the search for the Global IMMUNOTALKS account, and then you'll find a tweet that says ask questions here. And then you can reply to that tweet. And Xiaoyu's Twitter handle is at Xiaoyu Hu 13.
Yeah. Thank you, Kate. Probably I would reply to the tweets when I wake up.
Yeah. There's
next morning. The first thing next morning.
At your leisure. There, typically people will, they might send questions at different stages. A lot of people join the talks later at YouTube, so don't feel like you need to rush to do that right now. So yeah, go get some sleep, and thank you again for joining us. And I just want to remind everybody, next week's speaker will be Tim Hand, and we will see you then.
Okay, thank you. Bye. Thank you, Kate. Bye, everyone.
Bye. Thank you.